Okay, good morning, folks. And today we are going to be talking about translation. So, whether or not you watched my last video, it's whatever. But the last video discusses transcription, and so this one's going to discuss translation. So they kind of follow in sequential order. So if you're confused by transcription, feel free to watch my last video. If not, if you just want to watch the translation one, this is the one. So we are going to be talking about translation in depth, right? So kind of answering the questions that the other video that was posted on Brightspace kind of did not. And so let's get into it. So for those of you that this is the first video that you're watching, translation, just for a quick reminder, is the process by which we are going to go from that mRNA strand to the amino acid sequence, right? So mRNA, we discussed how that came to be in the transcription video, right? And so translation is the process by which the mRNA is going to essentially be read, and then kind of these amino acids are brought in by this other RNA molecule, but we'll get into it in a minute. And so that's the process by which mRNA kind of goes to this amino acid sequence, and amino acids are obviously, used to build proteins. That was covered in the last chapter. And so this is translation. So to begin, translation, similar to transcription, has three kind of stages. Again, like initiation, elongation, termination. But there's a couple of key players that we need to know before we get into those kind of stages. So what is a start codon, right? So start codons are base pairs on the mRNA strand, right? That are read, right, by the tRNA, right, that is going to kick off translation, right? So the start codon is specifically an amino acid called methionine, right? And that's going to be AUG, right? So those base pairs, right, that were kind of derived from the template strand of DNA. And so the start codon is important because it specifies where translation actually needs to start, right? So that's really important. And so how does translation know when to stop, right? How do those molecules, know when to stop? So you're going to have stop codons, just like you're going to have start codons, right? And so stop codons obviously are going to signal for translation to cease, right? To stop. And so there's three stop codons. Uracil adenine, right? UAA, UAG, right? Uracil adenine guanine, and then uracil guanine adenine, right? So those are UAA, UAG, UGA, right? So when tRNA is going to hit, that kind of codon block, right? If it has these three letters, right, you will kind of the tRNA will stop, right? And so I keep referencing tRNA, but what is it? So they are kind of these molecular kind of bridges, right, that are going to connect the mRNA codons to the amino acids that they encode for, right? So that's what tRNAs are going to be. And then you're going to have something called the anticodon, which is the end of the tRNA, which can bind to specific mRNA codons. So let's figure tRNA is my fist, right? So on this end, we are going to have the anticodon, which is going to bind to this mRNA strand And on the other end of the tRNA, that's going to carry the specific amino acid that is specified by the codons on this mRNA strand, right? So in review, anticodon, right? That's going to be this end of the tRNA. This is just where the amino acid is carried, right? mRNA strand. Bind, that will be read. This amino acid will be carried and it will be added to the long chain. But we'll get into more of the specifics of that in a minute. So the ribosomes are really important in the process of translation. And so for those of you that don't know what ribosomes are, right, because we haven't really gotten to the organelles section of biology in this class yet, but ribosomes, they're generally kind of known as the organelles that make proteins, right? However, the ribosomes, while they do this, right, this is a more of a specific description of them. The ribosomes are really important in translation because they are really where the tRNAs can find the matching codons, right, on the mRNA template and kind of deliver those amino acids, right? So the ribosomes are kind of like this big thing, right, where the tRNAs can kind of swoop in and the mRNA is right here, right? So they kind of act as this kind of conduit for what's going to happen. And so these specific ribosomal slots where this translation happens, right, are called A, P, and E slots and sites. You don't really, that's not particularly, it's just something to know, right? It's not like the main portion of translation, right? But just so you know the jargon. And so let's get into the specific steps, right? So first we're going to have initiation of translation. And how does this start, right? So the ribosome, first of all, is going to assemble around, right, the mRNA, right, to be read, right? And that's the coding strand, right? The mRNA that was produced in transcription, right? The ribosome is going to assemble around that, okay? And then we have that. Keep in mind, I should have also mentioned, okay, translation is going to happen in the cytoplasm. Transcription is going to happen in the nucleus, right? So two different places in our cells, right? So translation is going to happen in the cytoplasm. Anyways, so once this ribosome has assembled around the mRNA to be read, the first tRNA, right? So our first transfer RNA carrying methionine, right? Which is our amino acid, which is going to match, right, the start codon, right? So AUG, right? Remember, those are base pairs, right? So adenine, uracil, guanine, right? That's going to code for the amino acid methionine, right? Which is going to kick off translation, okay? And how do we know that these amino acids code for certain things, right? So in this biology class, you likely will be supplied with a chart, right, that will inform you of kind of like what these certain codons code for, right? This is not the case for all biology classes, but for all intensive purposes, this is an intro biology class, likely if you will be asked to decipher what a codon codes for, right? So what amino acids are being coded for, they will likely supply you with a chart. That is initiation. So ribosome assembles around the mRNA, right? And then the first tRNA carrying methionine, right, which is the amino acid which matches the start codon, AUG, is going to be carried into kind of this ribosomal space. That's going to start translation. Okay, next up we have elongation, right? Which is going to be when our amino acid chain is going to get longer, similar to how in transcription the mRNA strand is going to get longer, right? Because of the addition of base pairs, right? With elongation, the amino acid chain is going to get longer because figure the tRNAs are bringing these amino acids in, right? That will read the codons, right, on the mRNA. So figure you're just going to keep having amino acids kind of added in this process, right? So it's going to become kind of this long chain, right? And so the mRNA, right, something important to note, is read one codon at a time, right? So if we remember, a codon is three base pairs together, right, that code for a certain amino acid, right? So the mRNA strand is read by the tRNA, right, one codon at a time, and the amino acid that's going to be matching each codon, right, so based upon what the tRNA reads, right, on the mRNA, that amino acid obviously is going to be added to the growing amino acid chain, right? And so that's all that's going to happen in elongation. Okay? And so then we have termination, right? So termination is not as kind of complex as it is in transcription, right? In transcription, as we discussed in the other video, right? Transcription termination occurs, right, when you either have RHO independent or RHO dependent termination, right, which just kind of all depends on the protein factors present, right? But termination in translation is very easy. You just you hit a stop codon, right, so that tRNA is going to hit a stop codon. If we remind ourselves, that's going to be UAG, right, or UAA, or UGA, right, and that's going to begin when that stop codon kind of enters the ribosome, right, so when the tRNA is going to hit a stop codon on the mRNA strand, and that finished polypeptide chain, right, so that chain of amino acids is going to be released. And there you have it. That is translation. Obviously, if you have any questions, feel free to put them in the comments and I'll do my best to answer them. But yeah, I feel like the video was a little bit confusing in that it didn't really get into the nuances. So if you were curious about the nuances of translation, there you have it. So have a wonderful rest of your day, folks, and happy studying.